video, we're going to look at proteins, their structure and their function. Why I separated proteins out to the end in its own section is because both in the study of biology and specifically for this AP course, proteins are a big focus of testable questions. And there's a lot of cool stuff that go on with them, but they're very important in that regard. Proteins are one of the most numerous molecules in your body. Your body has tens of thousands of individual proteins of hundreds and hundreds of different sorts, and they're probably most important as enzymes. So they catalyze reactions or help different biological reactions take place that keep us alive for us. From getting energy from the food we eat, turning it into ATP, to different micro processes that go on in your cells. The functions of proteins. So over 50% of each of your cells by mass, dry mass, is proteins. And like I had just said in the last slide, proteins are active in every single process that goes on biologically in your body, both as direct participants and as enzymes. In fact, over 50% of the protein you eat in a day in food goes to making new enzymes in your body. If we take it to its most basic, there's 8 functions that proteins have in your body. Number 1 is their enzymes. So they catalyze reactions. For example, if we have an enzyme here, they might take this enzyme might take 2 sugar molecules, a glucose and a fructose, and catalyze that glycosidic linkage between them. A second important function is proteins are antibodies. So in your immune system, if we have a pathogen right here that's invaded your body, antibodies that stick to it, that surround it, and mark it for phagocytosis, like by white blood cells, like we'll learn about at the end of the year, these proteins, these antibodies, do that. Third, they're used for storage of energy. So if you think about in an egg, there's a protein called albumin that's in this egg, albumin, that helps to store energy for the soon-to-be growing animal that will be in there. 4, proteins are also hormones. So things like insulin is a great one that helps regulate glucose in your bloodstream and in your cells. 5, they are also receptors. So for example, if here is your cell membrane, it is studded with many different proteins on it that when stimulated by a specific molecule, maybe will tell the cell to do something specific. So what if this is endorphin? This receptor, this protein receptor, will trigger a cascade that will cause a dullness of pain, for your pain to dull the pain receptors in your nervous system. 6, they are used for contraction of muscles. So in your muscle cells, you have something that kind of looks like this. They are actin and myosin fibrils, and they're proteins that basically help your muscles contract and relax. 7 proteins are structural. So like collagen and things that form keratin, like our hair and our nails, use structural proteins. And 8 proteins are for transport. So they might move things through the cell membrane. So if this is our cell membrane, this might be a big transport protein that has a channel in here through which a molecule can go from the outside of our cell to the inside of our cell. Or we have hemoglobin, which is a protein which carries oxygen around to the different cells through the bloodstream. So proteins do a lot of different things. Just like all of our other biomolecules, proteins are made up of long chains of amino acids. So amino acid is the monomer of a protein. A bunch of these monomers, amino acids put together, is a polypeptide. That's our polymer. So amino acids are our monomers. There's 20 different amino acids that are in our body that we need, and they consist of 2 carbons. Here's a picture of an amino acid. The central one is called the alpha carbon, and each amino acid is the same in that it has a carbonyl group right here, or a carboxyl rather, and an amine group. That's what's called an amino acid, amine group. And it has this thing called the R group here. And this is composed of many different elements bonded together, but it's basically the only part that's different amongst all the amino acids is what makes up this R group. So these amino acids link together by dehydration synthesis reactions. So this OH group on the carboxyl side will form a dehydration synthesis with this H from the amine side, and they'll be removed to form water, and then this C will be attached to the N of the next amino acid. And a good example of that is if I open up this little animation I can show you, and we go to proteins. So here's 2 proteins, or 2 amino acids. Here's 1, here's 2. They line up like this, where we have the carboxyl end right next to the amine end of the other one. And when we go through dehydration synthesis, the water is removed and a peptide bond is formed between the carbon and the nitrogen of the 2 amino acids, and we're starting to build our polypeptide. All right. So here are all the amino acids, and we usually group them by R groups because they have different properties based on their R group. All of these sections are the same. That's our backbone. As you can see, the R groups are composed of various chains of different elements. There's some sulfur in some, there's nitrogen in some, there's double bonds, oxygen in some are really long. Glycine just has another hydrogen R group. And we group them roughly by non-polar side chains, so these don't have very many electronegative elements, or if they do, they're kind of don't create a permanent dipole. We have polar side chains, or hydrophobic ones. They have these, usually these OH groups hanging out there at the end, making in ends, nitrogens, making them very polar. And then we have electrically charged, so they have ions. Acidic ones will usually, when they're in solution, they'll look like this. They'll give up this hydrogen. So normally outside of solution they'd have a hydrogen right here, but in solution in your body they would give up that hydrogen. And basic ones that would in solution in your body gain and have a positive charge because they accepted a hydrogen at these locations. And those properties are very important because the R group properties determine protein folding. The R group interactions determine protein folding. So, polypeptides, which are a chain of amino acids, are not necessarily the same thing as proteins. The chain of amino acids is just that, a chain of amino acids. A protein is the shape that results. The shape that results. So, what do I mean by shape? So shape is important. The physical conformation of the amino acid chain shapes and determines how a protein works, specifically what it can bind to, like a lock and key. So for example, how a protein looks, its shape, determines what it can do. For example, if I had a protein that had a 3D structure like that, it might accept, or it might look for and bind to another protein that looks like this, that's on a pathogen membrane, and this protein might bind to that and release chemicals like cytokines in your immune system. So the shape determines what it can bind to, and the protein shape is extremely important because it tells us what the protein does. Well, there's 4 levels that determine the exact overall structure of a protein and its function. The first one, the primary structure, is basically in what order are we putting the amino acids. For example, here is the primary structure of one protein, and each of these circles is a different amino acid. If we change up the order of these same amino acids, we might use the same ones. If we change up the order, the primary structure is different. It's like combining the different letters, the 26 letters we have in the alphabet, to form all the different words and sentences we have. So we can arrange these in different ways to make different polypeptides, basically an infinite diversity of proteins. And most important, as we'll talk about in the next slide, the primary structure influences the secondary and tertiary structure. So small changes in the primary structure of a protein can affect a lot about its secondary and tertiary structure. Alright, the secondary structure is kind of created by the hydrogen bonds that happen in between the backbone functional groups of a polypeptide. For example, it's those coils and the folds that happen in the primary chain. So there's 2 types of secondary structure shapes. For example, there's something called the beta pleated sheet, where if we have a long polypeptide chain, it goes off the street and comes back, the oxygen double bonded to the carbon dioxide in one end of the polypeptide can actually form a weak hydrogen bond with the hydrogen on the amine group of a parallel part of that same polypeptide chain. And so they can kind of stick together like this, form these hydrogen bonds, these dots. So the backbone, the amine and the carboxyl group, are hydrogen bonding to each other to form these pleated sheets. In addition, the carboxyl group and the amine group can form hydrogen bonds in a different way to form this spirally alpha helix. So hydrogen bonding between the backbone of a polypeptide creates the secondary structure in beta pleated sheets or in alpha helices, an alpha helix. The tertiary structure, which is the overall, how does it look 3-dimensionally, is caused by interactions of the R groups. So those different R groups and their properties interacting with each other. For example, hydrophobic or non-polar amino acids are going to try to get away from solution, or in your body you're going to try to get away from the intercellular, extracellular fluid. So they're going to clump up close together like this on the inside. If this is a hydrophobic, if this is an amino acid with a hydrophobic R group, the R group is going to try to shield itself between the backbone and the extracellular fluid. So it's going to try to wrap itself up into this little oxbow like this. having the polypeptide background shield it. Hydrophilic, on the other hand, or polar, will want to be on the outside here. Those R groups will extend out here on the outside. Positive and negative charged amino acids will come together and try to form ionic bonds like this. And disulfide bridges between cysteine amino acids. Cysteine amino acids has an R group that looks like this. Well, if two come close together, these H's will leave and form elemental hydrogen, and the sulfurs will bond to each other, kind of drawing those two together and forming this big bow. So the secondary structure is caused by these different interactions between the R groups. And lastly, there's a quaternary structure. So some proteins have multiple subunits, which means there's more than one polypeptide that are linked together to form the chain or the whole protein. So there's a few chains that come together to form one functional protein. And this is called the quaternary structure. For example, here's hemoglobin. Each of these different colors represents a different polypeptide chain that each have its own unique tertiary and secondary and primary structures. But together, they come together and form one hemoglobin molecule. One hemoglobin molecule. What's really cool is that in sickle cell disease, changing one amino acid can change the quaternary structure of this sickle cell red blood cell. So normal red blood cells kind of look like this. They carry oxygen. And they have a specific amino acid sequence. In sickle cell, one of these amino acids that is hydrophilic is changed, and in sickle cell this one becomes hydrophobic. And what does that do? Well, now this hydrophobic amino acid wants to move away, get away from water. So if we're looking at a red blood cell, normally no hydrophobic regions, it's nice and round. Here, one of the subunits is going to have a piece that wants to migrate towards the inside of this globular structure, and you're left looking with a red blood cell that looks like this. And here is the hydrophobic region, the hydrophobic amino acid. So one amino acid can change the quaternary structure and the function of this protein, this hemoglobin molecule, because once it is sickle-shaped like that, it can't carry oxygen very well. Last but not least, what determines protein structure? Well, we've already talked about it. Interactions of side chains and backbone fold proteins spontaneously. So the interactions of the side chains and of the backbones through the secondary and the tertiary structure kind of develop how the protein is going to fold. But temperature, salinity, and pH can also break. If we raise the temperature, increase the salinity, and decrease the pH, can break chemical bonds and those interactions between the side chains and the backbone and denature the protein or cause it to fold in a different way. Also, if you think about it, if you change the polarity of a solvent from polar to nonpolar, it'll change whether the polar or nonpolar amino acid R groups are facing the outside or the inside. In addition, some proteins called chaperonins help fold giant proteins. They help in the folding process. In some diseases, like mad cow disease, can happen if proteins are misfolded. So for example, mad cow disease is a prion encephalopathy, is what it's called. And a prion just means a misfolded protein. It's not a virus or a bacteria, it's a misfolded protein. So in mad cow disease, proteins in the cow's brain that normally look like this get induced by a misfolded protein to change their overall three-dimensional tertiary structure to something like this, and they become misfolded. And where there is an alpha helix, there is a beta helix or beta sheet now. And that misfolding influences with brain and neuron function, and that's what also ultimately becomes a detriment to the cow. So that's it for proteins. I'll see you in class on Friday. If you have any questions about that, just give me a ring or an email, and I'll be sure to give you any help that you need.